Buonasera a tutti.
Good afternoon. My name is Gianmaria Zanella. I'm in charge of research and development at the Enologica Vason company. We'll talk about the improvement in the impact in enological practices and the synergy between products and technologies. And for us, the brand name is Aurora's. This is a claim that you certainly saw in magazines and journals recently. The point of my agenda is to focus on Aurora's and what it is. Aurora's is a brand by Vason Group, bringing together a number of products by Juclas and others by Enologica Vason in order to diminish the impact of enological practices in wine, giving us a product which is healthier, which respects raw material, and which is transparent and meets the requests for accountability. It is a philosophy, therefore. It is a philosophy which expresses the will of the group to devote its know-how to support the work of producers and winemakers, to transform the requests coming from clients into an opportunity for producers. And challenges for the future entail that the future already is today and is present today. We have a number of requests coming from the legislator about what has to be put on the label in terms of additives that are present. The origin of the request comes from consumers due to mass communication, which we have today. This was the effect, and so the regulatory boards followed the requests of clients. Of course, this is in line also with our philosophy for better sustainability for the future. We should use alternative products which are less impactful on the raw material, and this means using innovative production technologies and processes. Furthermore, producers should review their entire sustainability approach in the cellar in order to also optimize energy consumption. The sector benefits from the approach which is Aurora's, and we have the products under Aurora's that you see on the slide. These products manage to reduce additives or replace them entirely in wines. A number of them within Aurora's entail electrodialysis, which is ED for us, mastermind remove, dealcoholization technologies, MMR+, gas management technologies, and X-Pro. Now the details. Electrodialysis corresponds within the context of Aurora's to the desire to give guarantees in terms of tartaric stability without the need to resort to additives. Electrodialysis is not filtering, but it means applying a physical process whereby there is a difference in potential and there is a selective extraction of cations which are responsible for the tartaric stability. This also has an effect on calcium and not only on tartaric stability. This also refrains from causing problems related to the precipitation of calcium. Rather the opposite, it solves those problems. And as is the case with the cold stabilization, there is a better colloidal respect effect, which you see here. This is the same wine stabilized in cold temperature. The color is definitely different from the wine that we stabilized with our ED approach. Now, within the Aurora's project, we also have the dealcoholation technology, which is very topical. We started working on that at the beginning of the year 2000. Now, the membrane technology that we apply is highly respectful, and the driving force in case of dealcoholation is the extracting solution, which is outside the contactor membrane, whereby alcohol goes from wine to water. This is a process which occurs in ambient temperature. The costs are limited, both in terms of energy and of the system, which has to be set up. You respect the raw material, which is the wine, and you do not have an additional byproduct with the exception of alcohol. There is no evaporation of water. At Enoforum, we will also have a tasting whereby we give you the possibility to taste the outcome of this technology. And the same goes for this technology, where we have a management of dissolved gases with MR plus. We can manage the part in addition in terms of the management of O2, which is strategic, but there is more to that. We also modulate the CO2 with either an enrichment or an elimination, which is a lot, because if you have 200 or 400 milligrams of CO2, what that entails a major sensory difference in wine. And then it also removes H2S and methanethiol. It then has a function in relation to the cutoff of the membrane, which potentially eliminates free acetaldehyde. Now, when it comes to oxygen resistance, within Auroras, we have a specific and patented X-Pro yeasts. The inactivation occurs with physical processes at a low temperature in order to preserve all the molecules of interest so that they are not degraded, as would be the case in other methods. And with other methods, with X-Pro protection, we can prevent and cure the oxidation of wines. And with this, we can work so that we have protein stability. You see in the table that the very same wine treated with 5 grams and 10 grams per hectoliter has these data of proteins, TLPs, chitinase, while the Proteo test has different values. So we need to add less bentonite and to reach the same stability. What about challenges for the future? Well, there are many, of course. When it comes to X-Pro, we know that we should talk in a different way about the fining of wines. We talked about the X-Pro also in a previous edition, and it was a novelty at the time, but Xielab is now mentioned by all. We believe at the Vason Group that also when it comes to the fining of wines, we should change perspective as we did when moving to Xielab to assess color. We should not only stick to polyphenols or catechins. We should use dedicated X-Pro range products which can support producers in making choices. Another challenge for the future is for sure the enological management in the vineyard, an anticipated enological management, resistance inducers with microorganisms and deactivated yeasts. Here we have the GX tests, and the GT is the test, the reference. This molecule is very important in the defense against insects, but there is also a sensory effect in that it gives freshness. We have decided to continue working on this in order to reach a turning point also in the vineyard. Based on studies that we have been carrying out for years, we know that different microorganisms and different treatment times can indeed have an impact in increasing a number of aromatic classes. The time of treatment is paramount, as is the class of microorganisms. Now, to conclude, synergy for us within Aurora's is to work with producers step by step. We want to have fewer additives in enological products, fewer additives thanks to the use of physical processes. And this gives a value added. The connecting line from dot to dot is Aurora's. So this is what we want to achieve, a drawing that helps us bring all these aspects under one umbrella. Thank you very much. And thank you, of course, to the people that you saw on the slide. And I give back the floor to Piergiorgio.
Zanella.
Thank you very much, Mr. Zanella, for this overview of low-impact winemaking. Now the floor goes to Piergiorgio Comuzzo. Professor Piergiorgio Comuzzo will describe the biotechnologies of the future. Thank you, Emilio. Thank you, Vason. Thank you. I'd like to thank the organizers and the attendees of this conference. Now, when Gianmaria just opted me in for this, I decided to aim high. As my grandmother suggested, when you speak the truth, you can never fail. So I went back to the idea of biotechnology. The term was first used some 100 years ago by a Hungarian agronomist who referred to the production of commodities using and the processing of raw materials using organic processes. I found a definition of biotechnology that was relevant not because of the content, but because of where it was found. It was in a Convention on Biological Diversity by the United Nations, issued in 1992. In Article 2 defines biotechnology as any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products and processes for specific use. Now, this definition prompted me thinking on this concept, and therefore something came to mind, two essential concepts in my view. First of all, that humans use biotechnologies and have been using them for millennia. Let's think of all fermented foods. In the previous speech, we started reviewing productions from the Roman times. Now, wine, beer, bread had been known ever since the days of the Egyptians and Babylon. What's different between those biotechnologies and the biotechnologies we use today? First and foremost, the development of knowledge that was initiated in the past 200 years. And here I have mentioned some of the main, most important authors of that development. Jenner with the vaccination against smallpox, then Pasteur, Mendel, Fleming, and many others. What many of us, really many of us, think of when biotechnology is brought up, that is, genetics, engineered organisms, are essentially only the latest evolutions in biotechnologies. So humans have been using biotechnologies for millennia. A second key aspect is that winemakers today are biotechnologists who are highly specialized, who normally manage biotechnological processes. So biotechnologies and winemaking have gone hand in hand ever since the dawn of time. But this definition of biotechnology, I said to you, I liked it because it was part of a specific context. The goal of this Convention on Biological Diversity by the United Nations states that the conservation of biologic diversity, the sustainable use, and I wrote it in bold, sustainable use of its components and the right and equal breakdown of benefits from the use of resources and adequate transfer of the relevant technologies should support biodiversity. This is the connection that explains the direction that modern biotechnology is taking. So also at the societal level, the application of biotechnologies is increasingly perceived as an approach to reduce chemical inputs. And it's often mentioned in connection with the reduction of the use of sulfites in wineries and also aims at increasing quality and sustainability. As we shall see during this forum, research is strongly oriented in the same direction in terms of sustainable processing and low-impact processes. And also the policies and the funding of research is largely devoted to innovation in this field. The changes in production styles that we've witnessed in the last 20 to 30 years in winemaking have taken us from wine as a food to wine as a hedonic beverage. So if you add to that the issue of climate change and the fact that consumers are increasingly aware, attentive and sensitive to environmental and energy concerns, we will understand what direction biotechnologies are taking in modern winemaking. There are products that we're more aware of and that we know better in terms of their composition. We're moving from generic products to tailor-made products. I wrote my thesis under the supervision of Professor Cellotti in 1998. We were talking about tannins as a general adjuvant, but back then people were starting to understand that tannins are not all the same thing. So you have pips, you have tannins from different origins, and then the same applies to yeast derivatives. Nowadays we know that certain derivatives can start a fermentation, others that act as adjuvants for fining, and the others work very well in clarification, so they can be customized for different goals with the purpose of reducing the use of chemical additives. There's another development that we're seeing in scientific publications. These biotechnologies are often combined during production and for the use in the cellar with emerging technologies having a low energy impact. So that tells you at a more philosophical level what we're going to talk about. I will discuss this bringing case studies drawn from our research activity at the University of Udine in collaboration with Enologica Vason, and also certain very virtuous examples that I found in scientific reference, scientific literature. Now, research activities. We have started a PhD course with the support of Enologica Vason to understand, to obtain new concepts of yeast derivatives from non-Saccharomyces, and also the use of emerging biotechnologies. We studied these new concepts also via the use of low energy impact technologies to obtain the products, focusing on two goals: maximizing the production of polysaccharides and antioxidant molecules. Why the non-Saccharomyces? Because, as we've been told clearly by Professor Rumizio in the previous speech, and I absolutely agree with everything she said, non-Saccharomyces are a non-negligible component of lees. Many studies carried out in the past have studied Saccharomyces, but in lees, the proportion of viable non-Saccharomyces is generally high. Plus, there's a component that remains in the lees after cell death during fermentation. Certain strains of non-Saccharomyces can produce high amounts of polysaccharides, higher than Saccharomyces. Let me add to that certain non-Saccharomyces can also produce antioxidants. Take glutathione or thiolic compounds that are produced in certain non-Saccharomyces strains in higher proportions than by Saccharomyces. The first case study, the first example I would like to bring is the following. We've talked about tailor-made products customized for specific uses. It's now clear that the conditions for the growth of a microorganism and the conditions under which a yeast derivative is produced can define the characteristics of the wine we're producing. We have two strains here, Hanseníaspora and Saccharomyces, that were grown under the same temperature and ventilation conditions. The growth was carried on a medium, a model medium. We monitored the production of glutathione and antioxidants in general after growth and after the enzyme lysis induced via the addition of beta-glucanase. GSH is glutathione. We monitored cysteine or cysteine, and RPC is the fraction of the thiolic compounds associated with the wall proteins, skin wall protein or cell wall proteins. The two strains behave differently. Temperature does not seem to have a key role in the production of antioxidants, although in the case of Saccharomyces at lower temperatures we have a higher production of glutathione. But the key factor here in these tests was the presence of air. The two strains will behave differently. In the case of Saccharomyces, when there's air, Saccharomyces will produce more glutathione, while Hanseníaspora will produce more glutathione when there's no air. So the two strains behave differently. There's another difference here. We needed to consider both growth and enzymatic lysis for the next conclusions. Saccharomyces during enzyme lysis will produce more glutathione and cysteine. Hanseníaspora will do the same without air, but when air is present, it produces more cell wall thiols. So in the case of Hanseníaspora, if we induce a growth and then the lysis under aerobious conditions, there will be many thiolic compounds that can absorb oxidized compounds such as quinones. If you want to produce an excellent autolyzed compound, we will have to work without air. So the change in the growth conditions can yield derivatives with different functions. Another example is the combination of emerging technologies with yeast derivatives. That is how the use of emerging technologies can modify the composition of a yeast derivative. In this case, we have self-produced two yeast derivatives from the same strain. This was a Saccharomyces. The first was obtained by thermolysis, a key treatment to heat up the suspension of yeast at 120 degrees for 2 hours. And in the other case, it was produced using a high-pressure technology in the order of 1,500 bars. Both derivatives were added to Chardonnay at the maximum admitted dosage for yeast autolysates, and we compared it with the result of the addition of SO2 to the tune of 50 milligrams per liter after 1 and 4 months. Now that led to the development of color that was more similar to the color developed in the sample with SO2. So when compared to thermolysis, high-pressure lysis for the production of this yeast autolysate led to a better effect on color, and that because the high-pressure treatment at lower temperatures versus thermolysis, as you can tell from the table, made it possible to have more glutathione. Now, you might contend the following. Currently, yeast derivatives are yeast derivatives made from Saccharomyces, and that's all we can use. Well, in this example, I show to you how it is possible to sort of dope and change the composition of the lees in a different way. Now, we took an initial inoculation of Saccharomyces and plus a Hanseaspora and one with only Saccharomyces. We were not in season. We grew both microorganisms in synthetic must, and we separated the lees. One of the samples was treated with beta-glucanase and the other with high pressures. This was added to a proportion of 5% to a white wine, which was assessed after 3 months for various parameters. Now, what we can see here is that the combination of sequential inoculation and high-pressure treatment led, as you can tell in the blue rectangles, led to, first column is for color, a color that is less aged, so it preserved the wine from browning better than it was observed in untreated wine. In the wine treated, the lees obtained from Saccharomyces only, and also in the wine trade with sequential co-inoculation, but without the use of high pressures. And this holds true for the development of color. High pressure and sequential inoculation also better preserved the oxidizable fraction, because in this wine glutathione was more prevalent, just like in all the samples obtained by sequential inoculation, and then also a higher mannane content. Now let's look at the bibliographical references. We talked and we'll hear again about the non-Saccharomyces. We know what their problem is in general, the loss of the inoculate's viability. The Polytechnic University of Madrid, with a group of Professor Morata, has used high-pressure treatment on musts in order to drive down the microbial, the native or wild microbial load down. You see that both yeast and lactic acid bacteria are totally inactivated by high-pressure treatments. And you see the comparison with the sulfited mustard. This is combination of biotechnologies combined with the emerging technologies. What are the prospects? What further evolution are we going to witness? Research exists on this subject. I took another example from a paper, actually a review on chitosane. Chitosane is another biotechnology product that has potential and prospects. There are several papers that indicate how it is possible to use chitosane to support the immobilization of enzymes. What's the interest of doing this? We've heard about the regulations on allergens. The immobilized enzymes will do their function, perform their function in one, and then will be eliminated, leaving fewer residues. Pilot studies have worked well for lysozyme, for beta-glucosidase, the proteases for protein stabilization, or urease to control ethyl carbamate. The new frontier could be to work on molecular imprinted adsorbents, composite materials. In the reference I found, chitosane was being discussed, but we can think of others that have one characteristic, namely that they work as a mold and they can be shaped or molded so that the molecule will perfectly bind to them and be selectively removed. Let me now quickly come to a conclusion. Biotechnologies is a sector that is evolving fast. We don't know yet where things are going to in the future. For time considerations, we've only reviewed some potential applications of biotechnologies. We could have been talking about malolactic fermentation, bioprotection, nutrition management, and more. The future will bring new developments and knowledge in this context, in the context of sustainability, environmental sustainability, and energy sustainability. Obviously enough, something will bring to bear in research and something else won't. There are difficulties and prospects at the same time. Now, the hurdles, you know it. You are specialized in biotechnology and you know that there's no perfect technology. So there has to be a fine-tune of every technological application, and every technology needs to be adapted to the winery at hand. We've discussed the combinations between biotechnologies and emerging technologies. Some emerging technologies are more mature at present and therefore winery ready, such as ultrasound, and others are not so ready and so will require further developments and studies. Therefore, there are certain regulatory aspects that also need to be addressed. Saccharomyces cannot always be used as yeast derivatives. Biotechnologies, however, will remain a driving sector, a leading sector as part of the agri-food. And as Celotti keeps telling me, when we take a cup of coffee together, research goes on. Before I close, I would like to thank the Vason Group, Albano Vason, Dr. Zanella and Elisa Daipre, and the Udine University colleagues, especially Sabrina Voce, for working on this project with me. Thank you very much for listening. Thank you, Piergiorgio. I'd like to thank Professor Comuzzo. I'll now leave the floor to Federico Giotto.
From Giotto Consulting, and he will address us on new perspectives for the use of yeast derivatives. Good afternoon. My name is Federico Giotto, and I work in the field of consulting, research, and training and education in winemaking. Well, we worked especially with inactivated yeast. So this is a line of research that has been continued for a number of years now. This has also led to a patent, which is X-Pro, whereby you have a technique of inactivating yeasts at very low temperatures. It is called pasteurization, actually, which gives a product with a quantity of antioxidants that exceeds the quantity that you would have with a warm deactivation technique. This is a technique which is normally used also in orange juices, where thanks to the fact that pasteurization is not thermal, but using pressure, gives in the end product a higher quantity of antioxidants and vitamins. Then there is also dehydration technique, which is special. Now, I will stick to the enological part, and with a sort of hands-on approach, I will tell you what we have been doing throughout the research phases. The activity of the yeasts in the post-fermentation phase is well known. Less so in other cases and phases which are equally important, because yeasts can be very helpful also in other phases of vinification. And indeed, in this case, we initially have a protection effect of grapes and juices. Here we have a mixture of chitosane and inactivated yeast that gives a lot of opportunity in terms of color, which is what you see at the center. It is the color of the wine at the end of the fermentation. Chitosane also gives an antimicrobial effect.
Contro il Brettanomyces, contro la fermentazione di batteri lattici.
We know it from various aspects, but actually it has a very far-reaching range of action. So it can offer several opportunities against microbial content in grapes. One of the most interesting applications, I would say, pertains to the cold simulation technique, which is a technique whereby we can have more aromatic wines, which are more stable over the course of time, and where the effect is also rounder in terms of state. And this technique entails that the juice be put into very cold containers, and we talk about the juice of whites and rosé wines. It can remain then there between 6 days and 2 weeks. I recently came back from France, and you see the south of France by now, in the area of Provence especially, uses this technique in order to preserve color of the course of time in quality wines. Now, in function of inactivated yeast and chitosane too, you have a high antioxidant effect. In the traditional classic technique, you need to have a fairly high SO2 content, 20 milligrams per liter in juice, which entails that SO2 in the end, at the end of fermentation, is higher. While here with inactivated yeast, you have a powerful antioxidant effect. 10 milligrams per liter of SO2, free SO2. That is the quantity that we consider here, so much less. And it is a technique and application which proves very useful. If we consider this Pinot Gris case, we see that protection was provided by inactivated yeast with chitosane, and we then made the aromagram, we had the summary of OAV values, so the olfactory and aromatic values. We grouped them into the main sensory effects, which act as reference. In orange, you have the juice treated with inactivated yeasts, and there you have an increase in floral and fruity notes. It is a marked increase in that it was significant, and it is very important in Pinot Gris. Of course, inactivated yeast has the possibility to protect the juice. We have seen that. What about the situation when the inactivated yeast participates in the fermentation of wine? That was an equally interesting test, because it seems that this gives an effect during fermentation as well. We know that inactivated or deactivated yeasts also contains vitamins, and it seems that there was an interaction not only for color, and by the way, this is a Chardonnay test from 2021. We fermented the juice separate, and one part was added with an organic activating substance, while on the other in the other group we had the same plus the activated yeast. It is Chardonnay, and I would say that the most interesting thing is the aromas at the end of the fermentation. You see that there is a slight increase in beta-damascenone, which is a molecule that we know contributes to the overall aroma of wine. And what I find particularly interesting, and here we have Chardonnay and not Sauvignon, while some tests were also performed with Sauvignon with interesting outcomes. So what I found interesting is that we have the increase of both 3MH and the acetate form. Of course, this would deserve further observations. Clearly, it would have been easier to have an increase in the 3MH form, but here we had an overall increase in the overall aroma of the wine, especially with reference to passion fruit and grapefruit hints. Now, let's come to a fairly innovative concept, and it is a concept which can apply to reds. I've always been charmed by the fact that we can investigate the interaction of tannins and sensations. Clearly, we produce wine, and when we produce wine, we are happy to know that we obtained a certain effect in terms of tannins, but then we also have the sensory part. In order to understand that and to understand the sensory aspects of astringency and bitterness, we need to say that we have, of course, an interaction with tannins, and this effect derives especially from the interaction with salivary proteins. There are then other elements which are responsible for bitterness, because you see with the loss of proteins, you lose a part of lubrication. But as a matter of fact, this does not have an effect for a long time. On the other hand, there are types of tannins which directly act on a number of epithelial cells and also on mechanoreceptors on the tongue. On the right-hand side, you see three mechanisms which are interesting and which are important in that they clarify how fining and clarification in wines works We always thought that there were tannins there too, and that there was an interaction with proteins. But if we consider mechanisms when tasting a wine, we know that more complex mechanisms come into play. The first one is the interaction between proteins and tannins and precipitation of the compound. Then we have a second aspect, which is very important and which entails a ternary mechanism where you have proteins bonded to tannins, but also polysaccharides, which make the complex more soluble, which means that this has an effect on the taste itself. Then the third mechanism, which is fairly interesting as well, entails a competition between the protein and the polysaccharides because they also bind to tannins. I think that that is interesting, and it is one of the reasons why in a number of wines stabilize thanks to the permanence of the lees. These are a number of articles that we investigated and studied in terms of knowing about the interaction between inactive yeasts and the fining agents and the tannic sensations in reds. So clearly, this follows on important features. And we talked about inactivated yeasts produced at low temperature. And proteins that also stand for the clarification effects have a structure which is very much relevant when it comes to tannins. And if you have other temperatures, this tertiary structure disappears. So temperature is key here. In addition to that, as Mr. Comuzzo said, and many others, including the previous contribution, selecting the right strain of yeast is very important. Here on the left-hand side, you see several of them with their protein content, and then there is also the content in polysaccharides. Clearly, this needs to be further investigated, and we can raise the bar here and try and understand the mechanisms, because clarification, fining should be considered within a broader context. Now we see a number of tests of clarification, fining, with inactivated yeasts. And I'm not talking about proteins or protein layers only. I'm referring to the mechanisms I mentioned before, whereby polysaccharides also have an important role. And since I'm using up the time allotted to me, I would like to draw your attention simply to strain 4 and 5, because they are basically the same. You see that number 4 is green, and it is very interesting in olfactory terms and also for taste. 5 is black, and it is more central. No difference between the two strains. It is the same strain, but the difference lies in the way by which the yeast was inactivated. We studied that in order to understand that you can have a huge diversification based on the strain and based on the type of inactivation in the process of fining and of also identifying the distinguishing features of wine. This is interesting. I know I could speak at length about this, but I will skip a number of things. We mentioned rosé wines. They are very difficult to stabilize in terms of color, and that goes especially for rosé wines that are meant to be preserved for a long time. In yellow, you see a delta E value, which is the change in color between two treatments. In addition to using inactivated yeasts, as is the case with the 2 orange cases, we had then PPV and potato protein. This tells us that PVPP clarification, and which is one of the best fining agents, actually gives extraordinary opportunities to improve the wine. But oftentimes wines that were fined, especially if excessive dosage was used, oxidize more in the long term. There is a correlation here between a free SO2 and the yellow concentration in wine. And this is a study we performed about stability. And at the end of the study, we understood that wines treated with a specific protocol for inactivated yeasts had a free SO2 content, which was 6 milligrams per liter in the bottles that were analyzed. This is interesting, and here again we talk about the protection of wine. It is something that I presented back in 2019. You have 3 minutes left. In 2019, this is what we showed. You see in the right-hand graph that inactivated yeasts can protect wine against oxidation. And we suggested using inactivated yeasts also after protein stabilization, because each and every interaction with bentonite actually has an impact on glutathione as well. This is what we have after 4 years. We analyzed those bottles again after 4 years. On average, 6 bottles were analyzed, and you see that the effect of inactivated yeasts vis-à-vis oxidation is a stable activity over the course of time, which means that it is not simply an immediate reaction and then that's it. It lasts. We provide consulting both in the vineyard and in winemaking. So we know about the impact of climate change. We have wines with a high alcohol content today, and oftentimes phenolic ripening is not correct. Our agronomists have always used part of the inactivated yeasts. So with Vason, we performed a research about the various techniques and these inactivated yeasts. Certainly, this is one of the aspects that should be considered, and I'm referring to the fact that grapes treated with inactivated yeasts have more wood-like color of the seeds. This is then a piece of research that was done with CREA. We see that resistance is another topic that we should address. The joint research started 3 years ago. This shows what happens after treating with inactivated yeast. So the 2 molecules, pisaetanol and pisaid, are considered in this case. This pisaetanol is a fungicide. So this is what happens 2 days after treatment is given. A, B, and C are or have been treated with low temperature inactivated yeasts. The other one is an already registered product, and there is a major increase. So you see that there is a huge opportunity there also for vineyard treatment. Thank you very much for your attention, and I am at your disposal for questions, if any.